As most of you know, this is the Puget Sound Kelp Research and Monitoring Work Group. And the purpose of this work group is to increase collaboration and information sharing. And the goal is to be a diverse forum. And here's an interesting thought. We've done a 2-year pilot here. And next meeting, which is October 12th, will be the end of that 2-year pilot. So what I will be asking you all at the October 12th meeting is, do we want to continue this work group? Do we want to change it? So that's something to think about, and we'll talk about more in October. So then next item in the chat, could you all please introduce yourself and provide any updates you want to provide, and request any topics for future meetings? That would be great. And of course, we record this meeting and we post it on our website for anyone who misses the meeting. And so it ends up being a really useful source of connection for people. So I'm going to quickly review today's agenda, which all of you have. We're going to have brief updates from 4 people, and then we'll spend a half hour with Jordan talking about her recent publication along with all of the other co-authors. And then from 11 to 12, we're going to talk, people are going to share quick updates on upcoming fieldwork. So some people I've already talked to, they're on the list. If you would like to be added to this list to provide a quick update on kelp fieldwork you're doing this summer, just add your name to the chat, let us know in the chat, and Elizabeth and I will put you on the docket to speak after the folks that are already listed. And then finally, I'd like to pass on an announcement that Rieta asked me to pass on. You'll see at the bottom of the agenda, the Greater Farallones Society or Association has openings for 2 kelp restoration specialists. So that could be you if you'd like to apply for that job. It's open until May 28th. Okay, so with that quick introduction, I'm going to pass it off to the first speaker, which is Wendell Raymond. Wendell, can you share your screen?
Yeah, let me try here. Yeah, hopefully everyone can see PowerPoint. Okay, great. Thanks, Helen. Thanks everyone for being here this morning. I'm going to give a quick update on the development of the floating kelp canopy area vital sign indicator project. I work for University of Washington, but I'm working with a great group of people from Washington DNR, UW, Washington Sea Grant, Northwest Straits Commission, and the Salish Indian Nation. So thanks to them. So as you may have guessed, this project is within the Puget Sound Vital Signs program. And back in 2020, the partnership proposed a new indicator for the beaches and marine vegetation vital sign, that being floating kelp canopy area. So that is what we're working on creating. So a little overview of where we are in the project. Back in January, we released a scoping report and held a workshop. If you came to that, thank you very much. Your feedback and engagement is really appreciated. Since then, we've been looking at those comments, exploring data, and sort of building options for what this indicator could look like. In a couple weeks, we're releasing our options report, and that will be followed up by a workshop on June 7th, which I'll touch on again in a minute. Then moving into sort of the fall and winter, we'll be refining and finalizing the indicator with another report and workshop in December and January, and then a year from now, we'll have our final indicator product. So stay tuned for all of that. So to give a little background on the vision for this project, we're producing an indicator for the vital signs program, but this project really exists and touches on a lot of parts of the kelp plan. And sort of working within that framework, we really want the indicator to be a product of intentional outreach, diverse participation, and co-production of knowledge. And then thinking long-term, we really want this indicator to be a sort of central hub for information around kelp that can inspire action, both science, management, and outreach surrounding kelp to produce a product that's useful to a broad audience. So as we've been developing this indicator, we've been taking in perspectives and comments from a lot of different perspectives, from the Kelp Plan, tribes and communities and agencies. The Puget Sound Partnership has certain requirements, and so on. And so we've sort of synthesized all this together into a couple guiding goals and principles, and that is to produce an indicator that uses as much data as possible, engages with partners, and is accessible to a diverse audience. And this is all leading to what we call our proposed blended indicator. So I'll give you a little teaser of what the blended indicator is looking like. So we see the indicator existing or having two main pieces. One, summary visualization products, which are the things that you can go look at online, maps and graphs and visuals, but all of that being supported by underlying technical documents with lots of detail and documentation. And so I'll give a quick overview of what those summary visualization products are starting to look like and the hierarchical structure that they exist in, which we like because it balances, communicating quickly through the big picture take-home messages for status and trends in kelp, but also has more detail that people can dig into if they want to. So this structure looks like this. There's a big picture info map, and then this intermediate scale sub-basin pages and then sub-basin reports. And so the concept for the info map is a map of coastal Washington broken up by different sub-basins that have a kelp status and trend icon indicator that tells you, is the kelp increasing, stable, decreasing across different time spans in a given sub-basin. Then if you want more information, you can click on a sub-basin and that'll bring you to what we call a sub-basin summary page, which will have some key results, will outline the data used in that sub-basin. We'll have some plots and a breakdown of that trend icon. And if that's not enough information for you, that the sub-basin summary pages will lead you to a sub-basin report. So this will be a technical document that's structured similarly to a scientific paper with lots of detail on methods and analysis and many more plots and graphs on what's going on with kelp in the sub-basin. And importantly, this will include a rationale and justification of why a certain trend designation has been made. So that's a little teaser. Will our blended indicator accomplish our goals? Well, you tell us. In a couple weeks here, we'll be releasing a report that details many of these options, followed up by a workshop on June 7th, which kind of kicks off the comment period for this part of the project with comments due August 1st. And we'll put a registration link in the chat. Thank you, Elizabeth. And we had great turnout for our first workshop. We'd really love to have similar turnout for this next step. But also feel free to contact us to talk individually or with your organization on any thoughts or comments you have on the project and development of the indicator. We'd love to hear from you. So thanks.
Okay, so I did put that link in the chat, and I will take away your screen sharing so that I can pop mine up. Okay, let me pop this into presenter mode. What are you all seeing right now?
We're seeing we're seeing your slide and your notes.
Okay, so from current slide.
There you go. That's perfect.
Okay. Great. All right, so I will be brief, but I just wanted to revisit our update from the last meeting around Senate Bill 5619 and give you a little update on where we're headed with the Kelp and Eelgrass Health and Conservation Bill. So good news, the bill was passed and signed into law on March 30th. And just for those who haven't been following quite as closely, this bill will serve a important role in allowing DNR to address threats facing kelp and eelgrass within our waters through a couple of different measures. And so just a little background, this bill was put forward by legislators in 2021 in recognition of the important roles of kelp forest and eelgrass meadows in Washington state, including the very different roles, including mitigating climate effects, providing critical habitat, and also the cultural significance that kelp and eelgrass plays to our communities. And so the main pieces of this new health plan is that it will conserve and restore 10,000 acres of kelp forest and eelgrass meadows by 2040. And DNR is going to work collaboratively with a variety of partners to develop the kelp and eelgrass health and conservation plan, which is going to prioritize vulnerable areas and advance strategies for reducing stressors and minimizing impacts in support of that priority areas. And so I think what's really important to note is that while this is a collaborative process, we are very much in the beginning stages of developing what that engagement looks like. And so we will work with Max Showalter, who I believe is actually here today, and a robust team here at DNR to put together a community engagement plan that we will have ready to submit to the public and the legislature by the end of the year. And we have until December of 2023 to develop this initial plan with the map of the priority areas. And so we've got a lot of work to do, and we will continue to keep people updated as we move forward with that. But I'm happy to chat with folks offline about, the next steps that we're taking in order to do that. But what I also wanted to do was I'm hearing that we're using a lot of the same language. So kelp plan, kelp health and conservation plan, these all sound very similar. So I wanted to draw some distinctions between this part, Senate Bill 5619, and how it relates to the larger kelp plan. And so just really quickly, for those who are probably already familiar, in 2015, we had our eelgrass recovery strategy. And then in 2020, we had our kelp conservation and restoration plan. The kelp and eelgrass health plan actually advances goals that were identified within both of these two regional plans. So I've highlighted them in red here, but the big goals that this plan intends to move forward is to describe and document the kelp and eelgrass distribution and trends, reduce or conserve and identify areas for conservation and restoration, and also figure out which tools can be applied at those priority areas to reduce environmental stressors and support conservation and expansion. And so while this is aligned with the kelp plan and is really intended to advance key goals and strategies identified in the kelp plan, it is a separate plan. But what I want to also reiterate is that there is going to be very close coordination and alignment with the various efforts. So all of the work that's going into this health plan will be closely coordinated with these other initiatives that are already occurring to advance the kelp plan, including working with Jeff Whitty, the kelp plan coordinator, to track and then also just liaisoning between these various vital sign indicator efforts, the kelp policy workshops, and some of these other conversations that are happening so that there's strong alignment and that we're really building and advancing these things together. So we in October will probably have a more robust update for you on what that collaborative process is going to look like and where we're at with kind of beefing up our science team to address some of the needs. And we are happy to chat with anyone offline while we are kind of in between meetings. So I am going to stop my screen share. Let's see, where are you guys? You've all disappeared on me. Okay, and I think we're going to Jeff. So Jeff, if you are ready to share, you can just pull up pull up your screen share for me.
All right. Can you all hear me and now see my slide?
Yep, that looks good.
Okay, great. Thank you. Just to introduce myself, my name is Jeff Whitty. I'm with the Northwest Straits Commission, and I am the new Kelp Plan Coordinator. I met with you all during the last meeting to introduce myself and to introduce a couple tasks that have been set in front of me. I'm happy to say that we have started with some of these tasks, and specifically, we have set up an advisory committee for the coordination of the Kelp Plan, and that committee has met as of last week, in fact. The advisory committee I'm very excited about. It is a group of representatives from various agencies and groups throughout the Puget Sound and even abroad in California, Alaska, and Canada. And the backgrounds of these representatives include research, policy, and education, which are our three major components of the Kelp Plan, again, also known as the Puget Sound Kelp Conservation and Recovery Plan. With the help of the advisory committee, we are starting to move forward in prioritizing the tasks that will be undertaken primarily by myself, but also hopefully with the help of all of you. And so as we move forward, and I'm looking for help, and so I'm just reaching out to all of you to say that if you would like to be updated with the Kelp Plan or learn about involvement opportunities, to please email myself at nwstraits.org. And then also the other thing too is, since our last meeting, I've been holding a number of one-on-one meetings with a number of you. And I continue to do so to learn about your projects and what you all are undertaking in regards to kelp conservation restoration. And if you'd like to have a sit-down meeting with me, I'd love to talk with you. And you can please, again, email me at nwstraits.org. So that's my quick update for you all.
Great. And Dana, did you have slides?
I do, but I'm gonna pull them up in a sec. First, I want to just chat to all the squares on the screen.
Okay.
So I'm Dana Oster, the Marine Program Manager with the Northwest Straits Commission. And I'm also, you're going to be hearing the phrase Kelp Plan a lot. So I'm giving you a quick update on work that we started last year in partnership with Puget Charitable Trusts to look at. It's really supporting in some specific actions within the plan, and that is to strengthen connections between policy and science that we have in Washington State. During the development of the Kelp Plan, what we heard a lot from folks in kind of our resource management side is that a lot of the code we have, at least at the state level and certainly in the federal level, was a good start in protecting and restoring kelp. But the question really was, how are we using that language in the code and those regulatory authorities, and is it working? And so this work with Pew is really a follow-up to understand what's happening with kind of understanding the structure of the regulatory tools that we have, and are they or are they not being used effectively, and what can we do to help strengthen them? And so what we did last fall included a survey to many of those on the call and others to understand what information sources our regulators were currently using, what barriers were they encountering, and really what tools could we help provide, how can we connect them to both filling data gaps and information and guidance and resources for them to be able to do their jobs. In addition to that survey, we now have 2 workshops that will be happening this year, one in next week and another one later in the fall, and some topic-focused discussions, really diving into the details of how our regulatory world works to protect kelp. And I did want to kind of say, why does this connect to this kelp data group, Kelp Research and Monitoring? And that was because when we did the poll, and this is also part of our focus, we were asked the question about barriers. So what do you see as, I'm trying to talk and share slides, but that is not, here we go. What do you see as key barriers to make informed decisions in your position and to effectively implement existing regulations for kelp conservation and recovery. And the themes that we heard kind of across the board at all levels, local, state, federal, tribal staff, and others, was that the two biggest needs were really data gaps. So the data does not exist to evaluate the impacts to kelp, and data knowledge, not being aware of existing data that could be used. And this also connects to this group when we asked what specific information sources people used in their different roles. A lot of them included, so for instance, federal agencies, NGOs, and university researchers seem to rely pretty heavily on published literature as well as web maps. But when you looked at responses from state agencies and local jurisdictions, they were looking at internally kind of created resources a bit more and may not even have access to all this published literature. And so really at the end of all this work, the idea is we would like to get a sense of not only what do people need to know, but how do we get that information to them and in what format. And so that's the quick update on what it is we're doing. And I'll put my email in the chat if you have questions about this project or would like to find ways to get involved.
Great, thanks, Dana. All right, so we are actually a little ahead of schedule. Jordan, are you ready to go? Can we get you started early?
Yes, that sounds great.
Okay.
All right, let me share my screen. And Dana's, I mean, all of those intros were a great segue into this. Oops, sorry. Okay. Do you guys see the blue presentation?
Yep, that looks great.
Okay, great. Well, Helen, thanks for inviting me to give a chat here. A lot of the folks on this call contributed to this study either as authors or as some of the kelp experts that we used in our focus group. So this talk is about multiple stressors and kelp in the Salish Sea, and I'm focusing on a recent publication that, there we go. Oh, and to briefly introduce myself, my name is Jordan Hollarsmith. I'm the mariculture and macroalgae lead research biologist for the Alaska Fisheries Science Center in Juneau, Alaska. And this work was part of a postdoc I did at Simon Fraser University and with Fisheries and Oceans Canada. Thus the focus on the Salish Sea. I'm now focused a little more on Alaska. But the paper is towards a conceptual framework for managing and conserving marine habitats, a case study of kelp forests in the Salish Sea. And it is open access, so everyone should be able to read it, no problem. But if you have any issues, please feel free to email me. Here are all the great authors. Again, many of them are on this call. So part of the motivation for this work was that, as we all know, there is a widespread loss of canopy kelp in the Salish Sea. And as some of the folks who gave their introductions, there's a lot of data gaps when it comes to what is the extent of decline, what are some of the drivers of decline, what are these stressors that kelp are responding to. And so it's very hard to make management decisions when you don't necessarily have all of the information. And this is not unique to the Salish Sea or kelp. This is an extremely common problem for ecosystem management, environmental management all over the world, that decisions have to be made in the face of very limited to no local data. Especially because a lot of the declines we're seeing in ecosystems in the Salish Sea and around the world are driven by multiple stressors. Sometimes it's, a very specific stressor, an oil spill, and so you have to respond to that oil spill. But more often than not, it's got to be some combo of climate change, fishing, nearshore development, all sorts of things, and some likely have different levels of magnitude of effect on whatever it is that you're interested in. So what we did, using the Salish Sea as a case study, we wanted to see, can we make management decisions in the face of limited local data that are still robust. So our first step was to bring together a focus group of experts to create a diagram of stressors based on their knowledge of the system. So this followed the DIPSR model, kind of a simplified version of that. But basically we asked them, what are some of the indirect stressors known as drivers? What are some of the direct stressors known as pressures? And what is our state? What are we looking at? So for an example here, our state is bull kelp canopy. One of the direct stressors might be herbivory, and one of the indirect stressors that could influence herbivory is fishing pressure. So the experts, we gathered them in a room and by phone call for a few hours and kind of hashed out this diagram where they identified some of these bigger drivers, intermediate states, pressures on bull kelp canopy area. So you can see folks identified a lot of different things. And we won't, we'll talk more about them in a second. So then what we wanted to do was see what is the support in the literature for these pathways that the experts identified. So does the data available back up kind of what these experts determined based on their own intuition or observations, interactions with the system? So for the literature review, we simplified it a bit, got rid of those intermediate states and just focused on these pathways between the indirect stressors, the direct stressors, and the state. And so for all the literature, we identified or we recorded the pathway, for example, how shoreline development impacts salinity. We identified the location of the study. So was it in the Salish Sea or was it in a temperate coast somewhere else in the world? Our limit was temperate coasts, with the high latitude limit being where sea ice forms and the low latitude limit being the known extent of kelp. We recorded the results, specifically the direction of the relationship. Was it positive, negative, neutral? And we recorded the species for direct stressors to state pathways. So was it a floating or a non-floating species? And we also recorded the life stage for the kelp studies. So the results, there's a lot going on here. Again, I encourage you to look at the paper because we had a lot of, the literature reviews were extensive. But if we focus first on A, this kind of first one, this was the literature from the broader coast. So you can see all the lines are colored in, meaning we found literature supporting all of those pathways from this global literature. And the colors are the direction of the pathway. So most of them had some sort of consensus. Purple line means there was no clear consensus. Dashed lines means we found 2 or fewer studies. So, you know, not super robust, or you can't necessarily be super confident in that direction of the relationship. And a solid line is we found more than 2 studies. So in general, found lots of support. Now, if we move to the figure B, this was specific to the Salish Sea. And here we find much, much, much less research. This light gray means there was no literature that we could find that connected these 2 pathways. And then you see a lot more dashed lines, meaning very few studies. So support in the global literature for all these pathways that our experts identified, very little data specific to the Salish Sea, which is what we expected and why we did this study. And the results from if we look at just the pressures to kelp, so again, this is the direct stressors on kelp. Here we break it up into the global literature and Salish Sea and floating kelp and non-floating kelp. And if you just kind of step back and look at the colors first, you can see that there's a lot more blank squares in the Salish Sea that again reflects those gaps. And the numbers in the boxes are the number of studies we found. So again, in the Salish Sea, very few studies. And then especially lacking were studies on these understory species. And we know, I mean, we're focusing on bull kelp a lot because it's fairly easy to, you know, you can monitor it from the surface, but the Salish Sea has an incredible diversity of understory or non-floating kelp species. And for that, we have basically no scientific studies in the literature on those really important and diverse clades of species. And but again, just focusing on the colors, where there are studies in both the global and the Salish Sea, you can see there's the same color generally, meaning that the direction of the relationship is the same for these non-Salish Sea and Salish Sea studies, indicating that this global literature can be useful for informing us or making kind of informed hypotheses about what's going on in the Salish Sea, even if we don't have that same level of research. And then likewise for the life histories, this we kind of combine them all together. The adult is dark blue. So the grand majority of studies were on the adults, with many fewer on the zoospores, gametophyte, or juvenile stages. And this is important because different stressors might act differently on different life stages. There might be bottlenecks we're not aware of. So important to keep in mind that a lot of what we know is based on the studies on adults. So the major takeaways from this work is that we found high support in the global literature for the pathways identified by the experts. So this could tell us that their intuition about the system had high support in global literature. We found very few studies from the Salish Sea, but for where we did find studies, there was high consensus in the directional relationship between the Salish Sea and global literature. So that global literature might be a sound proxy for processes in the Salish Sea. And the majority of studies were on adult life stages of floating species. So we highlight some of the research gaps. We found very few studies on how development impacts nearshore abiotic conditions. And you think about the Salish Sea, this is Seattle, this is Vancouver. These are rapidly growing urban areas. And then also huge agricultural lands. It's in a rainforest, so you get a lot of runoff, a lot of connections between land and sea. But we found very little literature that actually looks at how those alterations to the land affect the nearshore environment. We found no studies on vessel traffic or dredging impacts to the abiotic environment. So you can see vessel traffic to all these links and dredging to most of the links are in that light gray. And again, Salish Sea has a bunch of boats, has a lot of ports that need to be dredged. So that could represent a very important gap for kelp that are found, along the near shore where a lot of people have boats or ports. And we found very few studies on trophic cascade impacts. And considering the huge alterations to trophic structures in the Salish Sea from humans, from long histories of fishing, extirpation of certain large fish species or otters, and just our knowledge from other areas about trophic cascade impacts on kelp, such as urchin overgrazing and things like that, it's likely an area that would benefit from increased research. Another big one was we really need more research on non-floating kelp species. It's logistically more difficult, but these represent huge and important or widespread and important habitats in the Salish Sea. And more research on emerging threats, such as contaminants or competition from invasive algal species could be very important moving forward. So the next steps of this work, Stacy McMahon is picking up where we left off for her master's work, and hopefully she'll be able to present to this group at future meetings, where she's taking the diagram that we created and the using our literature review to inform a qualitative network model. So this is just a kind of screen grab of what her model looks like. But the way this works is you can press perturb different nodes in this model. So you can say, what happens if we increase upland development based on our knowledge and of the direction of the relationships and how confident we are in those relationships. What's the cascading impact? And so she's able to test various scenarios. So she can perturb this model and say, what happens with climate change? So an increase in temperature or decrease in salinity from increased runoff. How does that then affect the whole model and ultimately full kelp canopy cover? So I believe, and she'll tell us more about it, she's testing perturbing all the indirect impacts, seeing how that changes kelp, perturbing the direct impacts, looking at a climate change scenario, and really interestingly looking at a scenario from the bill that Elizabeth was telling us about. So what would it look like if we had a kelp conservation area? What would that change? And then given the other stressors still at play that a conservation area wouldn't be able to impact, such as climate change, how would we guess that impacts full kelp canopy cover? So really excited to see where her work takes us. And so with that, I would love to thank all the amazing co-authors and the institutional support that led to this study, and happy to answer any questions.
Okay, so Jordan, if you want to stop screen sharing, we can pop into, we can see everyone's faces, and then feel free, I see some people using their hands, but yeah, if you want to answer or ask a question, feel free to either raise your hand or pop it in the chat. And Jordan, it looks like we have a first question from Mike. Go ahead, Mike.
Hey, I had a question. Has anybody taken this paper, and I know it's kind of new, and kind of reverse engineered it for a marine area analysis of what each one of these unique watershed or water bodies has as far as like a threat to existing kelp populations?
So that's a great point, and something that Stacy and I have talked about, because that would be another way to use the model that she's created, where you can say, okay, in this certain basin, there's a lot of upland development and warmer temperatures. So we can specifically kind of perturb those parts of the model and increase those and see how would we expect kelp canopy to respond. Is that answering what you were saying?
Yeah, I mean, I'll probably say this until I retire, but this process is very similar to the salmon recovery and the culvert issue, where all the people talking about it don't have a 2 or 3 dimensional view of the habitat, the species distribution, and the ecosystem that we're talking about. And I know things go from like a global to a regional down to a small watershed scale, but the work you guys put together in the pathway, it looks like all the MRCs, like from kind of Edmonds north, have the ability to do a rapid analysis of the strengths and weaknesses of their vegetative habitat. And that's something I would want to see the group look at, because you've got a diversity in the Salish Sea, north of Seattle, where their MRC work. You've got an existing body that's dedicated to increasing science in the Salish Sea. And you've created a system that within 12 months, working with House Bill 7619, you could actually prioritize, the wetted usable area or the lost habitat, and move the agenda forward to where people can start focusing research on basin-specific limitations to the kelp production. That makes sense.
Yeah, I think that's a great idea.
Other questions? Helen, I see your hand up.
Hi, thank you so much, Jordan. This is really important work. So thank you and all of your authors. Sorry, I have a cold. You noted there's been very little work on how development impacts nearshore abiotic conditions. And to me, this is a huge paradox, because it's probably the highest priority gap to fill in the Salish Sea. We're not going to stop development. So we really need to know what aspects of development are most deleterious and how those impacts can be minimized. And I'm wondering this, I'm going to ask you, but maybe this is an ongoing discussion for the whole group, is how can we figure that out? And is there other work addressing this question going on in other regions that we should look to, specifically general development impacts on nearshore abiotic conditions?
Yeah, so in answer to the second part of your question, we did find research from other regions, and our entire literature review is available as the supplementary materials. You can see what papers we found that went into these diagrams. So there is work going on in other regions. In the Salish Sea, we found some research looking at the linkages between upland development and contaminants, some on nutrients, some on sedimentation. That was mainly from the Elwha River Dam stuff. And then from shoreline development, some on sedimentation, some on temperature, but a lot of gaps. And I think what would be really interesting as a kind of a first pass is taking a remote sensing approach and just looking at across different watersheds, what's the ratio of different types of development, thinking about hard surfaces, urban areas, agricultural land, forest land, logging, clear-cut land. What's the proportion in different watersheds? And then what's the state of the kelp where the river lets out? And can we see any broad scale relationships across the entirety of the Salish Sea before diving in maybe to specific basin studies? This was something I wanted to do as part of my PhD work, and kind of how I originally got connected with a lot of folks in this group. Turns out it's pretty hard, and so I didn't get to it, but I still think it would be a really interesting study, and a lot of that information is available if you're a GIS whiz, which I never ended up being, so.
Other questions or comments for Jordan? Helen?
I'll add one more question that's keeps me up at night, which is this paper was really, really helpful. And to me, it sets the stage for the next step, which is thinking about these regions, these different stressors by subregion, because I think we all know that Puget Sound and the whole Salish Sea it's not, the environment is so heterogeneous based on where you are. So I guess what I'm wondering, I don't know if Stacy could do this too. Maybe Stacy could do everything, but it seems to me one of the follow-on needs is really to break down these stressors on a sub-basin or sub-regional basis. So anyone who's looking for work, we'd love that.
Yeah, that's why I'm really excited about this model that she's putting together, because, she'll focus on certain scenarios for her master's thesis, but then the model will be available for anyone to be able to modify and use. And it does come with some important caveats where the model looks at the direction of the relationship, but it doesn't incorporate relative magnitude of impact. So maybe a tiny change in temperature can have a huge effect, but a large change in nutrients has a relatively small effect. But the model doesn't account for those differences. It's only, does one change, therefore kelp changes up, down, not at all. So that will be, I think the model is going to be a great way to just kind of test out different scenarios, and then more specific work would be needed to really follow up on that. One thing I forgot to note is another kind of scenario that she's looking at for each is comparing areas with high stratification and low stratification, and how that might impact the ultimate state of kelp.
Okay. Other questions or comments? Hugo, you're off mute. You have a question?
Yes, thank you, Elizabeth. Thank you, Jordan, for your presentation. That triggers a lot of questions for me. And it relates to the connection of science and implementation of that science through different regulations. And one topic that I will suggest to explore is how effective our regulations are, and what do we have, the laws that we have, how effective they are in protecting kelp. It's something that I have seen firsthand, and if there is any kind of monitoring on that. Are we protecting what we are supposed to be protecting? Are those laws effective? And perhaps we could find a correlation to that. And so that would be, it's not a lot of science in there, but it relates to how we implement science into protection of ecosystems. So just an idea.
Absolutely. I mean, if, for example, the laws focus entirely on nutrient runoff or something like that, but we find that climate change and temperature changes are the big one, then is there a point to the nutrient law, you know, as just a broad example? And that can get really tricky really fast because, I mean, this group isn't going to stop climate change, unfortunately. And so there's, you know, that tricky balance of local management actions in the face of global stressors. So yeah, I don't know. We need to address climate change, obviously.
Other questions or comments?
And I think Stacy is on this call. So maybe Stacy, if you want to drop your email in and folks can follow up with you as well. And I will put my email in here too. So if anyone wants to chat more about this, I am happy to do so.
Great. Okay, last call. Any last questions or comments? It looks like we are having a little bit of a conversation in the chat. I'll direct people over there just to see what that's about. And okay, Jordan, anything else?
No, that's it. Thanks so much.
Okay, thank you. All right, we are ahead of schedule, which is great because that gives us lots of time for Tish. I see your hand just went up. Did you want to sneak a question in there?
I did. I actually just saw Eliza's comment in the chat, and I wanted not to put you on the spot, but that sounds really cool. Would you mind saying a word about that, a couple words about that study?
Sure, I'm out and about, so I don't have my video on. Let's see, it's something, I think it'll probably be directed to maps. It's still going through the NOAA review process, but we did dive surveys at 22 sites across Puget Sound, looked at understory kelps and other macroalgae, and then used the PPI index that he and Phil Levin have developed to look at correlations with urbanization. And I think what was most surprising was that urbanization came out pretty consistently as being a stronger predictor than things like salinity and temperature.
Can you define PPI? I'm sorry.
Yeah, so population proximity index, you basically just take a raster of population density, and then you're just looking at an averaged population density within a certain kernel radius or kernel buffer area, so to speak. We used a couple different scales for that, so everything from 1 kilometer to 20 kilometers, kind of getting at different potential scales of urbanization. We were hoping it's definitely a proxy metric. Kelp, though, I think 20 kilometers, the larger scale, was what was most strongly correlated there.
Cool. Thank you so much.
Yeah.
And Helen, your hand's up.
Yes, and so that sounds great, Eliza. And the thing that really strikes me as someone who's kind of standing between science and management is I think it's intuitive to many of us that urbanization would be a strong predictor. But I'm not sure how we turn that strong relationship into actionable management responses. So knowing that, which is, intuitive to many of us, what do we do next? And I'm really struck by that when I work with Dana and the DNR folks on trying to figure out how to handle wise urban uses. So, anything that we could do to further that understanding would be really great. And it's great to have the confirmation that our intuitive sense is right, that urbanization is a strong predictor.
Yeah, I just want to second that. That's also what I would love to love to see. It's such a hard thing to tease apart because there's so much, at least within the Salish Sea, there's so much collinearity between just general population density and all the other components of development that I think play into effects on kelp. I wonder if, and following this talk we just heard, which was super useful, I think maybe looking in other regions where there's less collinearity for maybe, guiding that process. It's tough, I think, just with the Puget Sound, though, based on what we saw.
And Eliza, we have a question, I think, towards you from Dustin. Can you point to particular urban stressors? So heat, imperviousness, nutrients, toxic shoreline impacts.
So we looked at, let's see, impervious cover, road density, and then just various land cover metrics. In the end, they all kind of perform similarly, and they're all interrelated. So yeah, it's just really hard between those to know what exactly is kind of the key piece.
Okay, thank you, Eliza, for jumping in. Any last questions or comments before we turn to our next agenda item? Okay, so we are going to transition now into our fieldwork roundtable. So we do have a few brief presentations already queued up. If you look at your agenda, you can kind of see who's on deck. So I'm just going to move in the order that is on that list. And really the objective here is just to share a quick update on what these groups will be working on this summer with regard to the field work. But as Helen mentioned, we did want to have this be an open conversation. So if you would like to provide an update as well, feel free to flag me in the chat. And then once we get through our updates, we'll have some time for open discussion, Q&A. We may wrap up early today. We certainly won't go any later than noon, but that is kind of where we're headed for the rest of the time together. So first up, we have Dan Abbott with Reef Check. Dan and Jackie, it looks like. So Dan and Jackie, are you with us and ready to go?
Yeah, we are here and
Perfect.
ready to go. So I'll go ahead and share my screen. Just kind of a quick update here. But yeah, I actually updated this group back in the fall. And I was looking back at my slides this morning to prepare, and this was my slide from last fall of what we're going to do going forward. And we have indeed done this. So over the winter, we hired Jackie, our coordinator. We finalized our protocols, and we developed a bunch of training materials, which brings us to the spring, which is where we are now. So Reef Check, we're community science. We teach volunteer divers. And so we're in the midst of a bunch of trainings. Our trainings are fairly involved. It's 4 or even 5 weeks of online trainings, video lectures, and then 3 days in the field. We do a minimum of 6 dives. Most of that is practice, and then everybody is evaluated. So far, we have completed 1 training and have 12 certified, citizen science Reef Check divers, with another 16 that are underway. We have a couple more classes planned that are coming up. So we're hoping to get some big numbers. I think our goal for this year is in that 40 to 50 range. So a big number. As far as what we have coming up for the summer, it's really to conduct surveys. So just a real quick refresher. We do transect-based surveys. So every site we lay 6 transects, core transects, where we count fish, invertebrates, kelp, algae, and UPC, and then another 12 transects where we just do fish. So each site tends to be about 14, 16 dives, a fair amount of work. And we're really looking at community monitoring. So, getting a kind of a good picture of the entire ecosystem. So we're looking at about 30 species of fish, about 20 invertebrates, 15 species of macroalgae, and in particular, a lot of that or non-floating kelps that I hear everybody talking about as being so important. Definitely something that we want to capture because we're down there, and because there's a data gap, and because I'm a phycology nerd, and so I look looking for that stuff. And then of course the uniform point contact, where we're looking at the substrate. Is it sand? Is it pebble? Is it reef? And all these protocols, they're different for Washington, but they're comparable to both our programs in California and Oregon, and to other monitoring programs in Washington. And we went through a big process to make sure that was the case. As far as our site location, so we're definitely still in the process of deciding where our sites are going to be. We have 7 index sites that we have committed to. And then another, potentially another, 30 or so sites that we're looking at. I don't expect us to get to all 30 of those sites this year. These are just kind of potential ones that we're looking at. But I do think, I think I said 12 plus. I imagine we'll probably be in that 20, maybe low 20s ranges this year. And I guess the point that I want to make is that we're really interested in a wide geographic range. So we're not focusing on any one particular area. We want, to have sites that represent all the different basins. And looking at both reef areas where there's canopy kelp, areas where there's not canopy kelp, and maybe just, subtidal or even just rocky reef, that places that used to have kelp. So we're really interested in kind of getting a big picture. And of course, that's just for this year. We are, our goal is just kind of ramp up and keep filling in these data gaps as we go forward. And of course, that is just our work. We are partnering with a lot of different organizations, and a lot of groups are, that we worked on our protocols with, are adopting those protocols, or at least very similar and comparable ones. So that from that, so in addition to the whatever, say low 20s surveys that we conduct, hopefully we'll have, other partners that are able to put together dive teams. Maybe we put them through a training and they're able to go out and collect more data in the same way. In particular, Puget Sound Restoration Fund is really our chief partner on this project. We've worked very closely with them, basically adopting some of their protocols and converting them for citizen science. But yeah, lots of partners. And that is it for me. Thank you very much.
Great, excellent. 1 or 2 quick questions, and then we'll move to our next one. Okay, we may circle back as questions come up, but for now, let's move on to Robin Fales with University of Washington. Robin, are you with us?
Yep, I'm here. I'm just gonna share my screen.
Perfect.
How does that look?
You might want to, yep, now it looks great.
Okay, awesome. So I'm Robin Fales. I'm a PhD candidate in the UW Biology Department and at Credit Harbor Labs. And I'm going to tell you about some of the work that I'm doing right now. And then you'll also hear from Brooke in a little bit that's going to talk about some of our other projects. Let's see. So I'm not going to give any background because I think we all know what our kelp stressors are. But our primary question is, does Nereocystis thermotolerance differ across the Salish Sea or with nitrogen content in the blade? And so we can think about this question using a thermal performance curve framework. So here's an example thermal performance curve with temp on the x-axis and performance on the y. The critical temperatures are at either extreme when performance is 0. And then the optimal temperature is when the performance is at its max. And then the habitat temperature is somewhere below that optimal temperature. And when we know both of those things, we can calculate the thermal safety margin, and we can also calculate warming tolerance. So we hypothesize that curves will shift across sites and with nitrogen content, where we expect lower nitrogen content to have lower performance and lower optimal temperatures and critical thermal maxes. So we're building these thermal performance curves across 7 sites with differing abiotic conditions. So here the inner diamond on these maps represents spring mean temperatures and the circle is summer temperatures. We have Squaxin Island and Tacoma Narrows in Puget Sound, and this is an interesting comparison because we know these sites are genetically similar, but experience really different temperatures due to mixing. We have Lincoln Park, which is warm and in Seattle. Cherry Point is also warm, but it's warm because of input from the Fraser River. And then we have 3 colder sites, which are Turn Rock, Tongue Point, and out on the Open Coast Olympic National Park. So at each of these sites, we'll perform a time search for intertidal kelp species. We're going to measure Nereocystis nematocyst size to get some size structure. And then we're also going to collect tissue for the experiments. So with that Nereocystis blade tissue, I'll have little pieces of tissue plugs, very small, and I'm going to incubate them for 36 to 48 hours at 7 different temperature treatments ranging from 9 degrees to 27 degrees at 3-degree intervals. So after that incubation period, I will perform oxygen evolution measurements to measure photosynthesis and respiration. We'll also do PAM fluorometry, and using dried tissues, we'll do stable isotope analysis and carbon to nitrogen ratios. Now for some very preliminary results from Turn Rock, which is a cold site. Here we have x-axis or temperature on the x-axis and FV over FM on the y-axis. And I fit a thermal performance curve to this data set. And with that curve, we can calculate the TOP, and that in this case is 15 degrees. The habitat temperature is 12 degrees, which we know from Kendall Murray's great data set for Turn Rock. And then the critical temperature maximum is 25 degrees. So the thermal safety margin is 3 degrees and the warming tolerance is 10 degrees. And so we'll be doing this and collecting this type of data across a bunch of different sites. And I'm excited to get the rest of that field work going. So I'd like to acknowledge my collaborators are Helen Berry and my advisor, Emily Carrington. And then this is funded by the Northwest Climate Adaptation Science Center Fellowship. So that's it for me. And I'm happy to take questions if there's time or in the chat.
Okay. A quick question from Steve. What's FVFM again?
So that is FV over FM is a metric that you can get using from PAM fluorometry, and it's dark adapted yield. It essentially tells us how well is photosystem 2 functioning. So it's a good proxy for photosynthesis. That's an instantaneous quick measure.
Okay. Any other quick questions? We have time at the end to circle back. Okay, thank you, Robin. Okay, we will now move on to Brooke and Sadie, also with University of Washington, I believe Friday Harbor Labs.
Awesome. Can everybody hear me okay?
Mm-hmm.
Great, okay. So I've talked to you guys before, so I'm just gonna give a pretty brief update on sort of some results from last season and what we're looking forward to for this field season. So I'm a postdoctoral researcher at Friday Harbor Labs, and I'm working on the effects of ocean warming and nitrogen limitation across the life cycle of bull kelp. And I wanted to introduce everyone to the newest member of our kelp conservation team at FHL, Sadie Small. So Sadie, maybe you can say hello briefly.
Hi, everybody. I'm really excited to join the group. Thanks, Brooke.
Awesome. Cool. So Sadie and I are working as part of the funding from the Washington State Legislature funding proviso. And we're working under the goal of the kelp conservation and recovery plan of trying to understand the environmental stressors impacting bull kelp in the Salish Sea. And so the overall goal is to identify environmental stressor thresholds across the life cycle of bull kelp. So we're specifically trying to identify thermal tolerance limits, like Robin was just talking about, and I'm collaborating with Robin on that, as well as nitrogen requirements for successful growth and reproduction of bull kelp in the Salish Sea. And so our group is doing lab experiments with bull kelp gametophytes, which are the microscopic phase of bull kelp that live on the bottom of the ocean, as well as with the adult sporophytes collected from multiple populations across the Salish Sea. And we're manipulating temperature from 10 to 22 degrees Celsius, as well as nitrogen levels across a range of ecologically realistic nitrogen levels that you might find in the Salish Sea. And I'm focusing on temperature and nitrogen because Helen Berry and others have shown that some of the sites in southern Puget Sound where we're losing bull kelp the fastest are those sites that have really high summertime temperatures, as well as really low dissolved inorganic nitrogen levels, because the water gets stratified and it gets warm and nitrogen depleted. So we're trying to examine the interaction between these two stressors. Just briefly, some updates from last field season. I found that gametophytes tolerated a surprisingly wide range of temperatures from 10 to 20 degrees Celsius, but we saw deleterious effects at 20 and 22 degrees. Basically, the gametophytes died. But importantly, completion of the life cycle, the production of those sporophytes only occurred below 16 degrees. So here's a graph from one of my experiments showing that you have sporophytes produced at 10, 12, 14, and 16 degrees, but not above 16 degrees. And so Sadie and I are repeating these experiments across multiple populations and with high and low nitrogen concentrations. And then additionally, I'm collaborating with Robin Fales on the effects of heat stress and nitrogen limitation on the adult sporophyte blades. So we did a preliminary experiment last summer exposing blades to 12, 16, and 20 degrees with both high and low nitrogen concentrations. And we found really deleterious effects at 20 degrees. The kelp had elevated metabolic demand. They had higher respiration rates and lower photosynthesis rates, suggesting that they're expending a lot more energy at 20 degrees. And then as you can see in the 20-degree low end treatment on the bottom right, we had significant blade tissue loss. The kelp blades kind of dissolved in that treatment, which was really bad. So we're going to repeat this again this summer across different populations that are naturally exposed to high and low nitrogen and temperature in the field. And then finally, I'm working on an informal network of subtidal temperature monitoring by deploying bottom temperature sensors at -3 to -5 meters relative to mean low water. And when we can, near surface or -1 meter temperature sensors. And these are all being deployed on existing buoys that have permits, so essentially mooring buoys. And so far there's sites ranging from southern Puget Sound that the DNR have deployed through some sites near Seattle, as well as a couple sites in the San Juan Islands. And this is just a few temperatures from last summer. You can see that Squaxin Island in southern Puget Sound is getting way warmer at the daily maximum temperature, upwards of more than 16 or 17 degrees, while sites in the San Juan Islands are staying generally around 12 degrees. And so the plan is to grow kelp from some of these populations where we have bottom temperature data, so we can pair the environmental thermal maximum with sort of the temperature tolerance of those gametophytes across populations. And then finally, I'm collaborating with Chris Neufeld. at the Kelp Rescue Initiative in British Columbia to look at population genetics of bull kelp from Washington through British Columbia and Vancouver Island. And the goal is really to look at connectivity among these populations across the border, as well as fine scale genetic variation using whole genome sequencing. So if you're interested in that, please look at Chris Neufeld's Kelp Rescue Initiative and get in touch, and stay tuned for updates. And then next, I'm going to show one slide from Kindle Miri, because Kindle is gone right now, but has been doing a lot of cool kelp research herself. So Kendall is a PhD candidate in Emily Carrington's lab at Friday Harbor Labs. And Kendall is really interested in asking to what extent bull kelp alter local seawater chemistry through metabolic processes in the Salish Sea. So Kendall has been diving and free diving to monitor a lot of sensors in the San Juan Islands. So she had sensors out last summer at Gordon Island, which is in the northern San Juans, Turn Rock and Harbor Rock near San Juan Island from July to September. She had moorings inside and outside of the kelp forest to look at seawater chemistry and pH changes with the kelp canopy. And then this summer, she's redeploying sensors at Gordon Island and Turn Rock mid-May through September. She's having extensive moorings at each site where she has sensors in the middle of the kelp bed, at the edge of the kelp bed, and then outside the kelp bed. And her goal is really to get at the effects of the kelp's metabolism on seawater chemistry, as well as how currents and tides impact that, because she found a lot of signal from currents, which are extreme in the San Juans, on her seawater chemistry from last summer. So with that, I think I am done sharing.
Okay, questions for Brooke? Looks like Steve's got a question. Anything known about temperature effects on spore production?
Yeah, that's a good question. So do you mean temperature effects on the production of zoospores in the kelp blade?
Yes.
Yeah, so that's potentially something that we might also try to do this summer, is to incubate some sori in the lab at different temperatures and see how the survival and viability of the spores and gravinophytes does. Yeah, I think we have a summer RU student working with us, and that was sort of the project we were excited about potentially doing with her, but we have to see what she's interested in too.
And then we have one more question from Mike, and then we'll move on. Go ahead, Mike.
Hey, I'm curious. So the Whidbey Basin has some very highly detailed, like 15-meter DEM temperature, salinity, and depth modeling done by UW and Battelle. I'm just throwing it to the group, is the data you're collecting for thresholds of productivity, they could be applied to the current data we're collecting in the Snohomish Basin. That would allow like probability of settlement of spawn. So the thresholds you're collecting are already being measured by the salmon recovery people. And I think the connection between those data that are like 20 years in the making in multiple models would be really good to apply to the limits to kelp production that you're seeing in the outside waters, outside the Whidbey Basin, that still have kelp, if that makes sense.
Yeah, so you're saying you're modeling temperature and other environmental variables around the Whidbey Basin, and then these temperature thresholds could be sort of put into your model once we find them?
Yes, I'm saying the data's there. It gets crunched every 5 years. The salmon guys keep doing it, and I see the money being spent. And I think the kelp community could really benefit from using the Whidbey Basin, which is missing kelp, and measuring water and salinity at a highly, like it's Parker McCready, UW and Battelle, and Terang. So the data is there to apply what you guys are measuring to kind of a wrecked system that's got some problems with kelp.
Yeah, that's a good point. I mean, the point of me doing these experiments is like to make this data available so people can use it for models and to inform management and all sorts of things. So we hope to make it published and publicly available as soon as we can. But yeah, I'm aware of Parker McCready's group and the live ocean model of temperatures. The problem is like temperatures vary a lot between sort of the mid-channel of the water and like directly in the kelp forest. So that's why I'm trying to sort of put out some sensors directly in the kelp forest, because we have a lot of good modeling data of temperatures, but we sort of really just need to know like exactly what the temperatures are in situ. But I agree that like the data could be used as inputs to the model in the future, which would be extremely useful and really exciting. So I super appreciate that thought.
Okay, so for the sake of time, we are going to keep moving, and then we may be able to circle back with additional questions, but feel free to also reach out to Brooke with the chat if you have any last questions. So we will move on now to Hillary and Gray with the Puget Sound Restoration Fund. Hillary, are you ready? Yep, I see you going.
Hopefully. Are we there?
Yep, now we are.
Okay, great. Thanks, everybody. I'm Hillary Hayford with Puget Sound Restoration Fund, and I'm sharing with you what our team's going to look like in the field this year on the kelp projects. And sorry, team, no one approved my use of these photos, but I wanted to show us in a joyful, natural setting. So also, I'd like to point out that recently Jesse Florendo joined our team, and we're expecting to have help this summer from Catherine Burnham. So thank you to Gray McKenna for prepping some maps for me to share with you guys, because they are getting massive GIS skills. And so this is a little more beautiful than what I could have created. I'm just going to jump through our major projects, beginning with kelp enhancement. So this is trial outplant enhancements funded by NOAA and the Washington State Legislature. And we've been working on this project for several years now. This is the third consecutive year at Doquigwitz, which is off of Port Madison. And this is inspired by comments from the Suquamish tribe about the kelp forest that was previously there. So what we've got is we've got 4 approximately 90-meter seeded kelp lines in the water that were outplant in February. And we included a trial this year of different seed densities so that we're working from aquaculture to lower densities to reflect that our restoration goals are really somewhat different than what aquaculture goals would be. We want to get a good, strong, reproductive forest that goes through the year. So we'll be visiting this site every 2 weeks after the kelp has hit the surface. Right now, we're just checking it below water as possible. And from the surface, we'll be looking at morphometrics and other sort of success and health measurements. We're doing a lot of work on the underwater kelp community monitoring network that we've been talking to you about over the last year. And we're planning to visit all of the 14 sites that are in this network, plus some additional sites this year. This, I just want to say that each of these dots represents the work that our dive team will be doing, as well as Reef Check, where they're overlapping with us, DNR, Point Defiance Zoo and Aquarium, UW Friday Harbor Labs, that's Katie Dubkowski's class, and the Seattle Aquarium. And of course, we have many other partners that we're developing, but these are the divers that will be in the water this year. This is, these 14 sites are funded by the Paul G. Allen Family Foundation on top of our existing projects and grants that I've already mentioned. And these will, the plan is over the next 3 years, each of these sites will have those annual biological underwater surveys, scuba-based surveys, but will also have a mooring with instruments on them. And the way that our moorings are designed is that there will be an instrument package off bottom and just below the surface waters. And so the work for this year is that each site with a solid circle is where we have biological surveys planned by us, our partners. And each site with a hollow circle is where we'll be exploring for next year's introduced surveys. And each site with a star is where anticipating that we can actually put out our instruments. This is pending permitting process that we're still involved in. Then we have a couple of special place-based projects that I wanted to highlight. One is with the Jamestown Sklallam tribe through a tribal wildlife grant that will be helping them to assess their intertidal and subtidal kelp resources. And there's an additional conservation and genetics component of that that I'll mention on the next page. Our other specialty project is continuing to work with the Port of Seattle on some habitat enhancements that have been done in Smith Cove that we are following, and as well as just helping them to catalog the extent of bull kelp throughout Elliott Bay. And then we have genetics projects going on here. This is primarily through a sea grant that we have with Felipe Alberto of the University of Wisconsin, the other UW. And this is building on past work that we did with Felipe a couple years ago to collect samples for gametophyte bank throughout Puget Sound. And what we're planning to do next is to continue collecting samples more extensively, especially along the strait. We're preparing for a reciprocal transplant that will be done next year, potentially at Doquigwitz and Freshwater Bay. And we're going to be working towards hiring the postdoc that's coming in to do the genomics for all of the samples that we have throughout Puget Sound and the new ones we continue to collect. And then again, with the tribal wildlife grant with the Jamestown Sklallam tribe, we're going to be adding gametophytes that we collect through their surveys to our gametophyte bank as we start to build that out at the NOAA Manchester lab. All right, that's what I got for you, folks. I know that was a lot of info, so please reach out to me if you'd like more details on any of those projects. And I'll check the chat here for questions.
Any quick questions? Feel free to jump in. Okay, we may circle back. Thank you, Hillary. Helen, you are up next.
Okay, I'm going to share my screen and show you a couple of quick maps. Let's see, do you all see a presentation mode?
Yeah, you just want to switch it so that we don't see the note side.
Got it. There we go.
Perfect.
Okay, I'm going to mention two of our ongoing kelp projects. We're scraping together all of our pennies to collect a large area of aerial photography that's really optimized for floating canopy surveys. So that's 6-inch or less resolution, ground survey distance, 4-band, and we're targeting low tide, slack current, clear and calm. And from these, we'll produce orthomosaics that will be available to other people. And I think some of you might find these useful. We'll also classify the canopies and use this data to update the vital sign indicator and other studies. I'm particularly interested in historical studies that we can use older data to move our baseline back from this 2022 canopy survey set. We don't know exactly how much funding we're going to have, so that our priority 1 aerial photography is in the yellow, and the priority 2 is in the mustard. And the way we pick these areas is just looking at the this northern Puget Sound has the biggest and the most abundant floating kelp canopies, so we're trying to capture those. As we also know that other regions are really important, so I'm going to jump down and look at Central and South Puget Sound. We've had challenges with mapping these beds with remote sensing because they're so narrow and low density. And so our goal at these sites is to continue kayak and drone monitoring, which you can really target at really precise times, high resolution. So you can see numbers 1 through 13. Those are the sites we have been monitoring. Some of them have absent canopies, so have been lost for either just a few years or decades. And then others have canopies present. you will also see we're trying to co-locate our data, our monitoring with as many other people as possible. And one of the exciting things is that at these sites 1 and 6, so Squaxin and Salmon Beach, the DNR dive team is starting to do underwater dive index sites similar to the work that PSRF and in concert with PSRF and also with Reef Check. So we're moving forward on this plan to co-locate different types of monitoring and also different types of research so that we can understand the trends we're seeing. So I'll stop there for now.
Okay. Any quick questions for Helen? Going once. Okay, thanks, Helen. Let's see, it looks like we are ready to move to Ian Miller and Steve Rubin. Are you two ready to go?
Yes. So I don't have sort of a well-formulated PowerPoint, but I think what we've got planned for the summer is simple enough that I'll be able to switch around to a variety of things and give you a sense for what we're up to. So first, let me just make sure, can everyone see I've got just a Google Earth up? Okay.
Yep.
So my name is Ian Miller. I'm with Washington Sea Grant, and I think Steve Rubin with USGS is on today as well, who's part of this interagency dive team that's evolved over the last 10 years or more at this stage. I'm not sure if Matt Byrne, who's with Lower Elwha Column Tribe, is also on the call today. I didn't sort of do a careful scroll through. But so we are planning for this upcoming summer, basically the same thing that we've been doing now for over 13 years at sites in the central strait of Juana Fuca. So that's why I wanted to start with Google Earth, just to locate everybody. We've got a set of subtidal monitoring sites around the Elwha Delta here in the central strait, and then a couple of what we refer to as control sites off of Green Point between Port Angeles and Sequim, or between Port Angeles and Dungeness Spit. So I'm going to switch in to a image to zoom in on our Elwha sites. Did that work? I switched to a static image here.
Yep.
All right, fantastic. It's a joy when it works. So we focus on 15 sites, 13 are clustered around the Elwha Delta. Those are what you see on your screen here. And then the 2 control sites off of Green Point further to the east. We've been surveying these sites, I think all of them since 2011. Some of them date earlier than that. So we've got 10 years of data at most of these and up to maybe 12 or 13 years at some of them. Some of these sites are also co-located with sites that were initially surveyed by the U.S. Fish and Wildlife Service, I believe in 2004. So we conceivably have, quite a nice ability to look back at habitat conditions at some of these sites. That is really kind of relatively unique in the Salish Sea, at least. So I'm going to switch now to a PowerPoint slide just to talk through methods a bit. So was that successful?
Yep. You might want to pop it in presenter mode, but otherwise it looks fine.
I will do so at the risk of.
Looks great.
Okay. Did that work? All right. So basically we use a modified PSCO methodology. Again, we sort of developed and adopted this in 2010, really. So again, we've got these 15 sites in the upper right. And then each site is marked with a what we refer to as a center post. It's just a stainless steel rod mounted on a heavy plate. So designed to be easily recoverable, although these have been out there now for almost a decade. And then we have a 50-meter transect that goes east and west off of each center post. So each of these sites is characterized this way. That's marked by a cement block. And we basically just stretch a tape between them. So each site is characterized by 2 transects, one to the east, one to the west. And along those transects, we're basically counting. So we're getting densities of algae, invertebrates, and fishes, benthic fishes, as well as bottom characteristics, substrate characteristics that we can analyze for variability within site. So we're tracking within 10-meter segments, or at the transect level, or at the site level, or at the region level. So the plan for this summer is to reoccupy these 15 sites again, which will give us, for a lot of these sites, more than a decade of data. I think that was all that I wanted to convey. Steve, was there anything else that I missed there?
No, that's good. But I thought we should mention that. So you can see here that we regularly put Mount Hobo temperature logger on the center post. So we have, and we've done this at the start. We did it at every site. And anyway, I just want to mention we have a lot of temperature data on the bottom at these sites, since that came up earlier. There might be some interest in temperature data. If anyone's interested in our temperature data, that's what we have. Thanks.
Yeah, thank you for mentioning that.
Okay, we do have a few quick questions. What's the source of the drawing that you posted?
The source of the drawing.
Helen, that was your question, so you might want to.
That was a question to Paul. There's been a lot of action.
Oh, I see.
Okay.
While you were talking, things are flying.
Never mind.
Sorry about that.
And Paul answered the question, which is one of their elders who passed away about 5 years ago, mapped some local kelp beds.
Oh, very cool.
Okay, looks like then Hillary's question was related. So do we have any questions for Ian? Okay.
I'd like to add one thing, which is I think some of the best long-term understory kelp dynamics data we have is from this work by Steve and Ian and others. And he did a Salish Sea ecosystem conference talk on this. So it's amazing stuff. And I'm really looking forward to seeing more data like this from PSRF and Reef Check and the other organizations that are starting to do dive surveys.
Yeah, and actually that, if I can ask a question after my own presentation, Helen, you, well, not only Helen, but I think others mentioned, you mentioned sort of the DNR establishing and occupying some index sites, and that term has been used before associated with the PSRF project. But is there an actual methodology at this point for those index sites that, I mean, I'd be very curious to sort of try to crosswalk between, the methods that we've established and what will be applied elsewhere, so that we can start to look at, where we can compare regionally.
I'll give a quick answer and then I'm going to punt it to Hillary, which is I think we're all trying to do protocols that are similar to yours. And Steve Rubin, when the PSRF was developing the protocols, we worked with Steve. So we're using the data sheets from PSRF, which, and I would love to put all the data sets together. I think this is the beginning of a network of dive sites for monitoring kelp and the rest of the community.
And so I would just add an update that, Helen, you probably haven't heard yet, that we met yesterday with your dive team to update them, because we've come together, Ray, Jackie, and Dan have come together on a Reef Check PSRF protocol, so that we'll be doing that across all of our sites. And as Helen mentioned, Ian, Steve was really instrumental in the way that we decided to do things in the first place, but there are some key changes because sort of the broad scale level is going to be a little bit simplified, which makes sense, right? So the select target species and stuff. I can share all that information with you guys later today. And then also, so everyone else knows, I just reached out again to Steve and Ian because we may be able to use our ongoing freshwater bay site at one of their sites that they already have this long record of data, and that will probably help us continue into the future to get that funded and get some more instrument data that's related to where we have all this biological data.
Yeah, and let me just add two other things that I neglected to mention. One is that we will be in the field the week of July 18th and the week of August 15th. Just if anyone, it sounds like there's at least one effort in freshwater bay, so just FYI there. And the other thing that I neglected to mention, but is hopefully obvious, is that the original site selection for our project was focused on understanding the impacts of the Elwha Dam removal on the on the coastal habitats, and so that really guided how our sites are located.
Okay, great. Thank you, Ian. Okay, we are going to move next to Zach Randall with the Seattle Aquarium.
Okay, can you all hear me all right?
We can, yep.
Okay, I'm going to share my screen. Can you see a PowerPoint?
Yes.
Excellent. Hello, everyone. My name is Zachary Randall. Thank you for the opportunity to speak to you all today. So I gave a presentation, I think last October, to this group, pitching the idea of a project, essentially. And I'm pleased to report that partially because of conversations that happened after that meeting, we have secured funding to bring me on board and formally initiate this project. So thank you, Helen, for hosting this group, and to you all as well for this great community. Okay, so what we are doing is developing ROV methodology for surveying nearshore benthic habitat, relatively shallow habitat. And first off, what we'll be doing, shout out to Kathleen Hurley with the Port of Seattle and Tammy Picorni with the North Pacific Coast MRC for supporting this work. There's been some talk about the impacts of development, and it just so happens that we're spooling up a project with the Port of Seattle looking into this very subject, not so much the impact itself of development, but more the patterns of association between benthic species along developed areas. So specifically, we'll be conducting surveys in Elliott Bay and trying to figure out what types of habitats, environmental conditions, taxa associate with bull kelp populations, with an eye towards what types of habitat and species associate with persistent kelp for outplanting efforts. For the North Pacific Coast MRC project, we will also be developing the ROV methods, though the geography is going to be very different, of course. We'll be operating out of Nia Bay along Cape Flattery and down along the outer coast. The field work will culminate in us accompanying the Olympic Coast National Marine Sanctuary and NMFS vessels when they go out and conduct their outer coast subtidal surveys this summer. We'll accompany them with the ROV and run the ROV along those diver transects. So that'll be a really interesting comparison between the two methodologies. We also have a lot of outreach planned for this project as well. We've scheduled a fair amount in May and June, so it'll be interesting to run the ROV for classrooms and community members, and they can view that live. A quick update on where we're at in terms of ROV development. We continue to develop and prototype the ROV. This actually isn't even the latest image. We've incorporated 2 more lights that face forwards. So in short, we have 2 cameras in the center here facing downwards, 4 lights facing downwards, and then 1 camera and 2 lights facing forwards. We have a sonar altimeter to provide a continuous feed of the ROV's altitude so we can acquire imagery at a consistent scale, and then a GPS tracking system to keep track of the ROV, and then later on overlay our data onto underlying habitat layers. So here, if it plays, I'm going to try to mute this. Well, this is some test imagery of the ROV. You can see a cryptic little friend here. We have, all right, I'm not sure if you heard the sound for that going through or not, but I moved on in case that was blocking the audio. We are able to get very good, crisp, clear imagery. We're still dialing it in, but we have been able to proof of concept both methods of analysis, the percent coverage and the abundance metrics. There's a lot of text on here. I'm not going to talk about this too much. I'm keen to explore next trade-offs between ROV altitude and the taxonomic resolution that we're able to obtain. So running the ROV at 1.5 meters versus 0.5 meters. I'm now thinking we might just want to get 2 different scales of imagery with reciprocal transects. In terms of analyses, we're all set to go with percent coverage from photos and then abundance metrics from photos and video. Since I last presented to you all in October, I've done a proof of concept with urchin-bearing imagery using Viami. So training Viami, this program, to detect what's an urchin with something on top of it, what I call a colored or covered urchin. That's the 2 different colors here. And so I can compile this into an algorithm and then apply that to the other half of the image set. So that worked really well for a proof of concept, and you can export data easily and work with it. And all this information is posted on a GitHub repository that's currently public that people can access. And I will be adding information to this in the coming days and weeks. I think that's all I have for you all to keep this short. Thank you very much for your support and for this kelp forest community. It's been great getting to know some of you, and I look forward to getting to know more of you and collaborating. So don't hesitate to reach out by email. Thanks, folks.
Thank you. Okay. Any questions for Zachary? Okay. Seeing none, I think we will move on to our last update. Just as a reminder, for those who would like to provide an update who weren't on our agenda, just grab me in the chat and let me know that you want to provide one out. In the meantime, we're going to move to Dana with Northwest Straits Commission to give us an update on their field work.
All right, you seen regular presentation mode?
I
am. To follow Zachary's presentation, I have an update on something equally as sophisticated in technology, and that is our volunteer-led kayak monitoring of kelp canopy. And so we don't quite have the algorithms that you have, but we have a team of wonderful, dedicated volunteers who continue to get in their kayaks with GPS units and paddle the perimeter of their bull kelp canopies that they've been monitoring. So this map here is showing you all of the sites that they continue to monitor. Many of them have started in 2015, 2016, and 2017 and go out every year, typically once per month from the months of June through September. And so this is just to show you the geographic spread of where we have volunteers in their kayaks monitoring bull kelp. And if you have kind of follow-up questions, really, I would encourage you to contact myself or Allie Simpson, who is our coordinator of this project. All of the data from the work that the volunteers have monitored is on our Sound IQ site, where you can look at the data up close and personal, and you are also free to use it by emailing Suzanne Scholl. She can compile it nicely in a GIS package to share with you. And that's all I was going to share.
Okay, short and sweet. Any questions for Dana?
I had a quick question.
Okay.
Do you have a particular app you use for your scientists who are out in the field?
We don't have an app. We have protocols on our website that I could put in the chat that they follow. And then we have a data collection system where they log data via Kobo Toolbox. And it's just basically an online web form version of our data sheet that they'll often take in the field.
Okay, thank you.
Okay, other questions, comments? Okay, well, I haven't seen anyone flag that they would like to provide an update, but I do want to open it up now for any additional questions or comments. We have a few minutes left for open discussion for anything you've heard today. So feel free to raise your hand or jump into the chat. While we're waiting, I am going to point Steve. There's a question, or actually it's from Steve to Dan Abbott. What is required to be a reef check diver regarding certifications, number of dives, etc.?
Yeah, for sure. So our minimum requirements are on our website. It's 30 lifetime dives, 15 of which are in the environment in which you're going to be working, and rescue diver certification. Those are the minimums. And yeah, I'm happy to reach out. We have public trainings that we're just doing for community members, but if you have an organization and a group of divers who you think would be interested in going through that training, we're happy to cater something directly towards that group.
Great, thank you. Other questions, comments?
Well,
Sylvan, I'm just
if I
Go
can,
ahead.
let me just use this as an opportunity as well. I may be jumping ahead of myself, but in regards to not having had a chance to talk to Steve about this, but you know, one of the things that we associate with this LWAD data set that I mentioned and talked about is that we've got this enormous, fantastic data set now that spans, you know, over a decade in some cases. But we have had limited capacity to subject it to analysis. And there's all sorts of stories in those data. So I just wanted to throw that out there that, that if anyone has that capacity, we are more than happy to share because we know that there's things in here that we just aren't going to get to.
Okay, great. Other questions? I'm going to direct folks who are, you know, using apps for citizen science programs to read Sylvan's question and respond to her there, as she's kind of crowdsourcing some ideas. Any other updates, questions, or comments? Okay. Helen, anything else you want to add before we wrap up?
No, I don't think so. Thanks you all for coming, and we'll see you in October. Have a great field season. Have a great summer.
Great. Thanks, everyone.
Thank you guys for organizing. Bye, everyone.
Thanks all. Bye.